So just quickly about me, I'm the Chief Manufacturing Officer at Brammer Bio. I joined Brammer about 4 months ago after a long career at Sanofi Genzyme, about 17 years. And prior to that, I had worked at BioReliance, and there's some folks you can see probably in the room that I may have worked with in the late 90s on gene therapy. And I also spent a bunch of time at a company called Wyeth in Pearl River, New York, working on vaccines. To sum it up, I'm an operations guy through and through. I've been through clinical and commercial production for many years. And what's exciting for me is really being back in gene therapy and seeing that it's really coming to fruition. I mean, this hope that was out there 20 years ago is now being realized in the approval of new products and the like. So I'm excited. And, of course, this topic in particular is becoming more and more critical as we think about how we're going to start to supply these products and ultimately supply them hopefully for lower cost of production so that we can get more access to patients. So with that, really I'm going to turn it over in a minute to our distinguished panel, but first let me just do some introductions of our panel. So starting on the right, we have Dr. Rob Kotin. Rob is the adjunct professor at UMass Medical. Many of you know Rob, of course, because he's been in this industry over 30 years. He's worked on AAV. And some of his major accomplishments have been the discovery of the DNA insert into AAV DNA, inserting into chromosome 19, as well as really vectorizing the non-AAV2 serotypes, which really opened the door for creating this sort of boutique of different serotypes we can mix and match for various tropisms and getting better efficacy of these drugs. And importantly as well, he invented the SF9 baculovirus process that many of the companies that work on viral vectors are leveraging today to make their products. So it's phenomenal to have Dr. Kotin on the panel. We also have in the middle Dr. Mark Plavsic. Mark is the Chief Technical Officer at Lysogene. He's the former SVP of Senior Vice President of Technology at Torque Therapeutics. And prior to that, he was the head of product biosafety at Sanofi Genzyme, as well as the head of gene therapy development during his tenure at Sanofi Genzyme. As a comment, Mark and I worked very closely together during his tenure at Genzyme, and one of the things I'd point out is if you know of the viral contamination we experienced in the Alston Landing facility back in 2009, Mark's forensics and detailed expertise on virology was essential to actually solving that riddle that we went through at that time. So I have enormous respect for Mark and his capabilities, and it's great to have him at Lysogene. And then finally, we have Dr. James Brown, who's the VP of Corporate Development at Aldevron. Dr. Brown prior to that, was the VP of Tech Ops at Regenix Bio, where he really focused a lot on contract vendor management, quality, kind of wore a lot of hats, it appears in his role, his past history. And when I talk to James, it sounds like that. And he has a Ph.D. from Stanford University. So we have a great panel. What I'd like to do is let each of the panel members sort of give an opening sort of perspective on where we see viral vector manufacturing today. And then from that, I'm going to target in on a few topics. And then I'll also open it up to the audience to really engage the panel with questions you may have that you'd like to hear a bit more dialogue and discussion around. So maybe with that, I'll start here with you, James, and maybe you can give a few thoughts about gene therapy manufacturing, where it's at.
Thanks, Chris. It's a pleasure to be here, and thanks to the organizers for giving us this opportunity. I think, as you said, there's just a lot of excitement now. Coincidentally, next year, 2018, is going to be Aldevron's 20th year in business, so as a company, we're pretty excited about that. And it's been great to see the growth of the industry and the things that are happening with the approvals that are going to be coming, I think, very soon. So I think when you look at the different trends, one of them is scale, so scaling up. So you see various CMOs and companies, CMOs like Brammer and Wuxi and Lonza and Cobra, and ourselves building new facilities or expanding facilities to supply the innovators and the companies with the materials that they need to make these programs, whether it's plasmids or viral vectors or things like that. So we recently broke ground on a 70,000 square foot building. We started looking, and Michael Chambers, our CEO, sort of had this vision of a green field. And as we all know, if you have that option in a new facility, that's great. So we're able to take advantage of that and get things up and running. we're scaling up to 300 liters and eventually 1,000 to 2,000 liters to supply material for this just rapid growth. And the other areas that growth is hiring. So, Chris, you're hired at Brammer. We hired a new chief operating officer, Henry Hebel, comes from 20 years of experience in plasmid DNA manufacturing, made one of the first GMP plasmid products for clinical trials. So it's great to have Henry aboard, and we're just growing our teams in order to meet the client demands. The other area really is there's scale up, make more of the same thing, and there's scale out. So with the advent of these personalized medicines, you have a situation where the product is very patient specific to the point where for us, we have to turn around and say a plasmid DNA from a sequence in a matter of weeks and have it be GMP. So what does that really mean? And it's a challenge that we are looking forward to. Michael saw this challenge a few years ago, and when he invented the GMP source quality system to provide critical raw materials for gene therapy and other uses, and he and the company are developing this vision of what that looks like, very small scale individual therapies, very efficient, and what does that facility look like? What does the process look like? What do the analytics look like for that? For example, sterility typically is a 15-day turnaround. Well, you can't really wait for that when you have these things, so what's the strategy there? And the other area is when you look at where we as a company sit in the supply chain manufacturing plasmids and proteins, typically our material will go to another manufacturer. It might be someone doing a formulation. If it's a direct DNA product, it might be someone using it to manufacture a viral vector or cell therapy. So we are really trying to work better with folks like Brammer and make this really a seamless supply chain. So if you're the company that needs the material, you just want it to work. And in some cases, for us, our client is the CMO, the downstream CMO. Sometimes it's the direct innovator, the sponsor of the trial, but we have to get our material to the CMO. It has to meet requirements that they have. So really trying to work with that group to understand what it is that they need. In the case where DNA is the API, it might be someone doing a lipid nanoparticle formulation or some other formulation. So how do we make that process efficient? And then if you look at, that's sort of the material after it leaves us, the material coming into us we're looking at as well. So how do we manufacture bacterial cell lines? We're looking at developing our own bacterial lines in order to improve the process as much as possible. And there's a lot of emerging technology out there with better plasmids, whether it's mini circles, nanoplasmids, these things that reduce the extraneous DNA that you have at the end. So we're looking to enable those technologies and partner with people in that space. So again, we can make improvements and have this seamless supply chain. And to that end, we're looking at off-the-shelf products. So we've manufactured Cas9 at the GMP grade, and that's going to be an off-the-shelf product. So some of these plasmids that are common to all of the manufacturing, can we do something where we have those off the shelf so we can reduce costs and timeline for customers? And then our clients are thinking about commercial a lot more. So they're looking at long-term supply agreements. They're looking to lock in pricing, better supply chain management, reservations and guaranteed capacity far into the future. And we're seeing them interested in tighter services agreements with assurances about freedom to operate. A lot of these licensing cases, the license will extend up to commercialization, and then you have to go back to the inventor or the whoever owns the IP for a commercial license. So how does that work? And then on the regulatory front, I think people are struggling with, how much process characterization and validation do you have to do with these different components along the supply chain? So I think there's a wide variety of opinion out there. We've seen clients be very conservative and spend a lot of money in that area. Other clients look to optimize their supply chain and say, where can we save time and money? So it's a real balance there. I think that with DNA at least, the process isn't necessarily the product. You can characterize it, you can sequence it. So I think there are more opportunities there to look at the process and develop it and not have a big effect for your downstream manufacturing. And then, along those lines, we're looking at process improvements and what do we need to do from a regulatory standpoint. So if we change a cell bank or if we scale up, what are the requirements depending on where we are in the supply chain? And we're finding the answer is, so far, pretty good in the sense that if we're a raw material for a viral vector, we can do things like possibly change the bacterial cell bank, change the conditions, or even the construct itself. If you want to move to something else like a nanoplasmid or optimize the backbone, as long as the gene of interest obviously doesn't change, you can do that and maybe not have to do a lot of comparability, maybe a small animal study at the most. As you scale up, you obviously have to do cleaning validations on the larger reactors, but beyond that, you have the opportunity to move very quickly. We're looking at also, dedicated spaces for campaigns, so we'll have a situation where all of the equipment and staff for a given campaign will remain the same throughout that campaign. And I think to end, it's all about hiring and retaining talent as well. So we're actively recruiting. We have a lot of open positions on our website. There's a lot of growth in the area, a lot of scramble for talent. So we're trying to position ourselves to attract and retain that talent so we can meet these requirements as we move forward.
Okay, well, thank you. Mark, you have some thoughts?
Yeah. Good morning, everybody, and it's a really great pleasure to be here this morning. In preparation for this seminar, I was specifically asked a question to discuss today about, dose planning, clinical, commercial entrance, and really translation from the development phase to the commercial phase. What are the challenges there, and what to consider when moving from early to late to commercial type of things? The key question is about transition and how to address that transition and make sure that there is a sustainable, uninterrupted supply of drug to patients. So thinking along those lines, when we speak of the early type of developments, it's important to keep in mind this in the phase 1 type of trials. These are very first patient access drugs. The process that needs to be developed needs to be well designed for the phase, needs to be commensurate with the phase itself, and it should be quick. And the key goal there is to make sure that the product actually works before going any further. So from that perspective, typically in a phase 1/phase 2 trials, what's important to consider is number of patients that needs to be done, and making sure that actually that we understand also dose for those patients. Speaking of the dose, it's very, very important to do the very clear calculations in terms of what is the total dose per patient that needs to be administered. And from that perspective, understand what is the concentration of a drug per vial. Understanding that, understanding the volume per vial also play an important role, because all of that plays has an impact on manufacturing supply and making sure that sufficient amount of drug, sufficient number of vials are supplied per patient. So in that type of exercise, it's important to understand those type of elements and really translate exactly into the phase 1 exercise. Sometimes, depending on the type of trial, the phase 1 actually can lead to phase 3 directly, and beyond that to commercial. So in some cases, depending on the type of trial, if the trial is going to be an adaptive type of clinical design, then in order to avoid later upsets and comparabilities and delays, it's important to think from the very early phase beyond phase 1, so that you start with the technology that is going to be right, and then as you move from phase to phase, as you approach commercial, you just build on it, provide more characterization data on the same platform, and just keep the same platform without changing. More and more I see lately that companies are engaging into an adaptive type of clinical design, in which case, really these things need to be considered very carefully. In cases where no adaptive clinical trial design is needed, and we call classical way of making sure that manufacturing process is going to deliver what you need and is compliant with the regulations that are designed with the phase 1 trial. As you move to the phase to the clinical late stage developments and also commercial, it's important to think a little bit differently. In the late stage development, one needs to keep in mind commercials as well, because there is a potential opportunity there, that phase 3 will work, and beyond that, you have to move the same platform into the commercial business. So from that perspective, commercial thinking and planning is important, and also making sure that we minimize the number of changes beyond phase 3. So to really maximize on the time, minimize on the cost, and also minimize on the regulatory hurdles as well. So from that perspective, again, going back to what I said earlier, if you think clearly and sufficiently enough on the phase 1 trial, it may actually lead to right design to phase 3 as well. The key question in the late stage trial is actually, can the manufacturing technology actually meet the clinical and commercial dose demands? So again, clinical planning and commercial planning plays a critical role in designing a manufacturing technology and manufacturing scale. Selection of the right manufacturing technology is essential, because that is a technology that is likely going to enter the commercial stage, and technology that one needs to live for a number of years beyond. And scale as well. Question becomes, is it going to be scaled up or is it going to be scaled out? And from that perspective, one needs to be very careful about planning which technology to select and how to manage the technology in the actual manufacturing setting. So all those planning elements we discussed earlier would apply here, with one additional point to consider, and that is post-phase 3, post-BLA, market access. They're really planning carefully about launching the product onto market and understanding the potential patient population out there that one is going to treat, and more specifically understanding how many patients are going to be approximately treated in the year 1, 2, 3, 4, 5 post-launching, and also understanding what is the peak going to look like in the product life cycle, so that that information can also be very influential in selecting the right technology and understanding the scale as well. Oftentimes, people make mistakes at this point, and oftentimes we hear or see in the news manufacturing issues, and gene therapy has a problem with manufacturing and all of that. It is not that we don't know how to make the product. It is actually planning and selection of right technology and translation of right technology into right clinical and commercial platform. That's where the mistakes are made, and that's where the manufacturing comes into trouble. Thinking manufacturability in the phase 3 or late stage development and commercial is important, and from that perspective, one really needs to ensure that process development for the phase 3/commercial takes into account some very important elements, such as for the right platform and the right scale, simplicity is important. Making sure that product is going to be operated in a simple manner without too many complications and opportunities to fail. The robustness and characterization of that process also need to be well understood, so that one really needs to understand if the product process works, why it works, and if process fails, why it fails. Oftentimes, process will fail if it's underdeveloped or poorly characterized or non-robust, and then it leads to a number of deviations, number of investigations, costs, and certainly issues to supply to market. So addressing those elements and preventing these issues at that stage is really important, and that's where the robustness and good planning for development plays a critical role. Certainly, economy and kind of cost factors, cost of goods sold, are important to consider for the scale in the product as well. Therefore, thinking along those lines is absolutely important to understand economical sustainability and things of that nature. We spoke earlier by James about raw materials and starting materials. I would probably say understanding really critical components of the process, such as starting materials with cell banks or plasmids in gene therapy, and critical raw materials is essential. Oftentimes, plasmids are coming to be a bottleneck because people oftentimes misunderstand or underestimate the quantity and size of manufacturing and supply of plasmids. And that is not a trivial process. Making plasmids is a manufacturing process for itself. It can be as complicated as making an AAV vector. And it needs time, it needs big scale, it needs sustainability, it's costly as well. So planning that and making sure that the supply is in place that cannot jeopardize manufacturing process is of the paramount importance. And I think I just want to corroborate what James stated earlier, This should not be underestimated. When speaking of late-stage development and commercial, building in the whole concept, regulatory and quality compliance and current regulatory and industry initiatives is also a very important consideration. There is a kind of a current dogma in manufacturing process which goes like this. Quality target product profile, CQA, CPPs or critical process parameters, process controls, manufacturing. So this is a flow chart, that kind of drives how process should be developed, and this is really what agencies like to see. This is embodied clearly in the recent ICH guidelines document, and compliance with this is important, because these will need to be clearly outlined in an IND submissions and all of that. So things like process control strategy documents, quality by design / design space, and safety by design, oftentimes neglected. Safety by design basically means product and process needs to be safe from the adventitious viral perspective. QBD doesn't necessarily address safety by design. Therefore, incorporation of adventitious agent safety in the whole process, from the raw materials throughout the whole supply chain to the end product, is also very, very critical to avoid later upsets. Finally, a couple of more points to make. Late stage, commercial, that's the stage where we need to think about platforms. If one is dealing with the same vector or the same type of approaches in the company, platformization plays an important role, because it will help manufacturing planning, it will help manufacturing plant, infrastructure, and simplicity, as we talked earlier. From that perspective, we can think of platformization on different level. Cell line platforms, plasmid or / transfection platforms, manufacturing upstream and downstream platforms, and probably I would go further and say formulation platforms. That also would lead to something very important, that is analytical platform, because many assays have become bottleneck in the future. So if you apply platformization across the supply manufacturing process chain, you will capitalize on a number of common elements and avoid issues in the manufacturing process. And finally, the manufacturing business model. Do you do it in-house? Do you outsource? Different companies have different models, and they need to be respected. Selection of a CMO, if that is a model to be followed, plays an important part. So having a CMO who is competent, who is knowledgeable, who has done this, who is very responsive, very flexible, and open to thinking forward in the commercial level is very important. Along those lines, I would like to say one more thing, that unlike several years ago, today we have one element in the whole gene and cell therapy which we didn't have back a few years ago, and that is what I called infrastructure. Infrastructure basically means it's an ecosystem that comprises the following elements. It's manufacturers / sponsors, it's CMOs, and CMOs are very important because a few years ago we had only a few of these. Today we have more. We have more to choose from and more quality CMOs to do. The CROs, which we haven't had many years ago, now we have much better situation. The suppliers of starting materials, plasmids, CMOs there, we are having much better situation today than a few years ago, so we can pick and choose those that actually can deliver for the phase 3 and commercial in terms of quality and sustainability. And also contract testing institutions, which we have a little bit more of a situation today. So putting it all together, we are in a relatively good position today to deliver and have a product on the market a few years ago. It is not to say that we are in a perfect spot. We are not. We still have room to improve, especially across the CMOs and CROs, but definitely it's much more comfortable situation today. With that, I'm going to conclude and turn over to somebody else.
Thank you, Mark. Rob, you have some thoughts?
Yeah, I do. I think James and Mark covered just about everything that I would have described. But so I'm not a biotech person by genetically, so I'd like to ramble on a bit as an academic and having a long perspective, a long view of the AAV field. So there's no problem AAV manufacturing AAV viral vectors. So that was said 25 years ago, and obviously there was a problem, but it was the publicly disclosed attitude of many biotech companies that were around then. They didn't really have a scalable manufacturing, but they represented they did. And over the years, not much has changed. People have gotten better at making vectors using conventional approaches. The quality control and analytics have improved, but the process tends to be inherently non-scalable. So the few scalable processes there are involve a system in which the manufacturing ingredients are amplified in the same system, so that secondary, tertiary, et cetera, cells in that process are then competent to produce the vector, not just the cells that are transfected with a plasmid or a non-replicating virus.
Sorry, I'm having difficulty hearing you. Yeah, I'm bringing a mic. Sorry, Rob. Bob is listening, so.
Won't be able to. Is this better?
Just a little closer. Yeah. Should be on.
Better? So I don't know if anyone can.
Well, just try.
Yeah. Go ahead.
Just
Okay.
speak. Yeah, just yeah.
All right. So developing of large-scale economical production processes have improved somewhat, but mostly there's no economy of scale. Transient transfection is just expanding the number of cells that are transfected. It's not increasing the total volume of cells that are.
Being closer to your mouth.
What's that?
Okay. No. Yeah? Okay. So as Mark had said, having the CROs, CMOs have gotten better over time, but yet there are no shortage of these companies that have never done this before and may or may not succeed. That can set a company's agenda back years or even put them out of business. So David Dismuke and I are working on a manuscript about what are the obstacles for clinical gene therapy using AAV. And we realize it's not the lack of facilities. There are plenty of GMP facilities in the country. Many of them are mothballed or underutilized. What is lacking is know-how. So that is the limitation in producing AAV vectors. Finding the CRO to do the process development or CMO who actually knows what they are doing in this process to ensure a high probability of an outcome to keep the trials on track and not have these frequent failures that have occurred. So again, 25 years ago, speaking to we just developed the SF9 production platform. I was speaking to a very recognized figure in the field who university, and he asked me what do I see as the biggest obstacle of transferring the technology from NIH to elsewhere, and I said the ability to follow directions. and no matter how detailed the protocol is, you cannot list everything not to do. And no, I shouldn't be surprised any longer, but people are very creative in figuring out what shouldn't be done. And it could be, and you cannot list everything. So, and so the CMO would say, "Oh, we followed the protocol. It didn't say not to do that. No product or poor quality products." So these get more and more detailed and more robust. But basically, the robustness comes from the experience. So then the quality of the vectors. I think the different groups doing different QC assays will report different outcomes. So having an experienced person in-house or a consultant or at the CMO who has done this before, who knows what are the important criteria, what are the for the FDA, but also for the as a unique biological, which AAV is. It has 3 capsid proteins, a simple linear single-stranded genome, and then it's supposed to have biological activity, which is accessible or saleable using a variety of methods. There's other aspects, physical aspects about the particle and the formulation, stability, monodispersity, etc. But basically, it's the composition of the capsid that determines how active the particle is. Another confounding area of the field has to do with the trend to find the next best vector, the next best capsid. The capsids that are available now work quite well, and the hope is that there's one that will be even better. So if the gene therapy program's not succeeding, it's not because it's not the best vector available. There's never going to be the best one. It's because we're not using the ones we have properly. And you can look at the years ago, the cystic fibrosis gene therapy field. For a decade, it was convincingly demonstrated that neither adenovirus or AAV were suitable vectors for that, but that kept going for quite a while. So the tools, these vectors are tools. They have appropriate uses and inappropriate uses. Maybe the new serotypes, new capsid variants will resolve some of these issues, but right now it's more like winning a lottery. It's 1 in a billion, 1 in something chance of finding that. So again, the manufacturing of each of these new capsid variants, even though they overlap with each other and the same know-how could be employed, it is a different product. So the downstream process has to be developed for each of these, and the turnaround time is not days or weeks, but it could be months for that to be done in terms of a GMP compliant process. So I think with the availability of the reagents that have been in use for either transient transfection, the SF9 baculovirus system have been well distributed, a lot of experience with a number of manufacturings, and yet there's still good producers and not so good producers. So again, I think it's incumbent on the end user to do their own diligence and validate the batch records of these service providers before engaging in their services. So
that's it. That's great, Rob. Thank you. So I think we just hit on, obviously, there's a lot of complexity. We covered a broad range of topics. What I thought I'd do, we don't have too much time left, is offer it up to the audience if anyone has any questions for our panel that they may want to ask. We have our wonderful cube here that will be thrown at you if you have a question. I do have a quick hit question for the panel here, but I'd love to get the audience engaged. You have a question? Oh, sorry. I thought you were waving at me. Okay. I guess the one thing I'd ask the panel, and I'm going to look for a quick response here, is if you were going to name 2 to 3 things you think are essential when you talk about the speed to go from, let's say, the vector core lab to commercialization, that idea that you're at phase 1, because a lot of companies create a construct and they quickly want to go into the clinic and get into commercial. What do you say are the 2 or 3 elements you really don't want to change that are critical not to change at that early phase through commercialization? Maybe I'll start with you, James.
I think clearly obviously the gene of interest, and you'd be surprised, well, people don't change that, right? But what we'll have is clients take 2 or 3 candidates through to a certain point, and then as they sort of narrow it down. So making that decision sooner rather than later is very important. And I think, for us, when we talk about capacity, it's forecast. How much material will you need at what quality level when? And to the extent that clients can do that, it just makes the whole project and the management much easier.
Great. Mark, any thoughts?
For me, yes, I agree. With the gene construct, if that can be maintained without change, it'll be fantastic, and certainly cell banks.
So locking in the cell bank and the construct is the theme I'm hearing. And Rob, any thoughts on that topic?
Yeah, right. The vector genome is critical. The downstream process should be substantially the same as what's been done during development. And, yeah, of course, the cells, all the reagents have to be what's been used during the process development, tox studies, and so on.
And maybe, so again, I'll offer it up to anyone in the audience that may have a question for the panel. I think oh, please. We'll get you the cube. Hold on so everyone can hear you. You got to speak into the cube.
Talk into the cube, yeah.
It's not just there to keep you company.
Oh, it really is.
Yeah.
It's just so there's a growing split between the camp of baculovirus manufacturing believers and mammalian cell manufacturing believers. Curious if you have any views on that. And then separately, we do see a lot of the innovators building their own manufacturing facilities. And so what is the role, or how should they be seeing the role for a CMO in the field? Thanks.
So, can you repeat the first question?
I guess the merits of baculovirus manufacturing versus mammalian cell.
Yeah. So the merits are that the virus, baculovirus, is replicated in the same cells that we're making the AAV. The AAV vector genome is rescued from the baculovirus genome, or it could be a stable provirus. It's amplified to very, very high copy number, just as if it was a productive AAV infection with a helper virus in a vertebrate or mammalian cell line. So we're recapitulating the natural biology of the AAV. The vector then is, the capsids are made at very high amounts. We tend to get, we have the capabilities of getting 70-80% filled capsids with no fractionation. The downside is that we have to use engineer the capsids, the cap genes, to get expression of VP1, 2, and 3 from a single ORF, and that often has to be changed to increase the amount of VP1, which determines infectivity. Getting a sign that says running out of time. There's a lot of nuances to it, and there's been a lot of bad vector made in SF9 cells, so we're always trying to clean up those messes. But during 3 years at Voyager Therapeutics, they were able to produce an AAV2 for the Parkinson's disease program that was comparable to the material produced at CHOP that was used in the clinic, purity, activity, and so on.
So, thank you, Rob. Mark, do you want to maybe comment on the make versus buy question that I think was asked? It was something around the choice of build your own or contract out. As a CTO, Lysogene, what are you thinking about in that context?
Again, as I mentioned earlier, different companies have different attitudes, whether to outsource or to keep it in-house. I can speak from the perspective of the company I work for, Lysogene, which is a virtual type of company, phase 3 and phase 1 as well. We opted to outsource. That's our current business model. It doesn't mean it won't change, but certainly for now, that is it. So we rely on CMOs, and therefore selecting a right CMO plays a critical part in our business. And there is a whole selection process which we can go through if you want. I don't know for the sake of time whether we have sufficient amount of time to go to that. But again, it's a really business decision and direction that each company has to establish for itself.
Thank you, Mark. So maybe what we'll do is I think it's time to kind of one more question. Okay, can we take one more question? Does anyone have a question out there?
I actually had one.
Oh, please. James,
I was interested in, you talked about manufacturing meet demands. How do you view shelf life? In other words, the stability. Are you actively trying to figure that out, or do you just say, hey, beyond a year, we're just going to make some more? Or how do you kind of maximize the amount you have from a lot that you can use over time?
That's a question for me?
Yeah.
Yeah, so that's a important point that needs to be also considered, apparently. Stability is managed through the stability testing program. When we develop formulations, we have to develop it in mind that can actually give the product shelf life for more than 1 year. Preferably 2, 3, 4 years or so. So that's a process that needs to be, it's a science that needs to be incorporated into formulation testing and stability testing program as well.
That's great. Yeah, and obviously it's the FDA and the EMA will definitely want more formality.
Sure.
That includes plasmids, by the way, as well.
Yes, it does. Yeah, we've actually, as people get closer, we're having more and more people do stability, right? As you might expect.
Yeah.
That's a good point.
So there's a lot we didn't get into today with our distinguished panel. Clearly, this is the first workshop of the conference, so Rob and James and Mark will be available all week, and I encourage you to talk to them about some of the many important topics they brought on the table. We could have definitely made this a full-day session for sure. So I want to thank our panel for doing an exceptional job here, and appreciate the audience for engaging and participating in this. So we're going to transition now over to the manufacturing of gene-modified cell therapies, and Dr. Jessica Harmon will be the moderator for that. So again, thank you, Rob, Mark, and James. Let's give a round of applause.